Hi, and welcome to BioLearn Academy. Ever wondered how scientists can get so much information from a minuscule amount of DNA? Maybe from a single hair found at a crime scene, or a tiny blood sample? Or how diseases like COVID-19 can be detected so quickly?
The secret lies in a powerful technique called PCR, or the polymerase chain reaction. Think of PCR as a molecular photocopying machine that can specifically find and duplicate a tiny segment of DNA millions or even billions of times.
What is PCR and why do we need it? So PCR stands for polymerase chain reaction. The name might sound complex, but the idea is brilliant. Imagine DNA as a massive library full of books, the entire genome, and you're only interested in one specific paragraph within one specific book, a particular gene or DNA sequence. The problem is, you only have one copy of that paragraph, which is usually not enough to study properly.
PCR solves this exact problem. It finds that specific paragraph, the target DNA segment, and within just a few hours makes millions of identical copies. This amplification makes the DNA segment abundant enough for scientists to easily visualize, study, and analyze. This revolutionary technique was invented by Kary Mullis in the 1980s, earning him a Nobel Prize.
The ingredients for PCR. The key players. To perform PCR, we need a few key ingredients. Let's meet the main players in this molecular drama. 1. DNA template. This is the original DNA sample containing the target sequence we want to copy. It could come from blood, saliva, hair, bacteria, or any biological source. 2. Primers. These are short, custom-made pieces of single-stranded DNA. They act like bookmarks, binding to the specific starting and ending points of our target DNA sequence. They tell the copying machinery exactly where to begin. 3. Taq polymerase enzyme. This is the star of the show. It's a molecular building machine. Its job is to read the DNA template and synthesize a new DNA strand using building blocks. Crucially, this enzyme is heat-stable. It comes from a heat-loving bacterium called Thermus aquaticus. 4. Nucleotides, dNTPs. These are the actual building blocks of DNA. Adenine, A, guanine, G, cytosine, C, and thymine, T. The Taq polymerase uses these free-floating bricks to construct the new DNA strands. 5. Buffer. A solution that provides the optimal chemical environment, like the right pH and salt concentration, for the Taq polymerase enzyme to work efficiently.
Okay, with all ingredients mixed, we place the tube into a machine called a thermocycler. This machine precisely controls temperature, cycling up and down through a programmed series of steps, over and over again. Each PCR cycle involves 3 main stages. Step 1, denaturation. The temperature is raised to about 95 degrees Celsius, 203 degrees Fahrenheit. This high heat causes the 2 strands of the DNA double helix to separate or melt apart, like unzipping a zipper, creating 2 single-stranded templates. Step 2, annealing. The temperature is lowered, typically between 50 to 65 degrees Celsius or 122 to 149 degrees Fahrenheit. In this cooler environment, the primers can bind, anneal, to their complementary sequences on the single-stranded DNA templates. Remember the bookmarks finding their spots. Step 3, extension, elongation. The temperature is raised again slightly, usually around 72 degrees Celsius or 162 degrees Fahrenheit. This is the optimal working temperature for our Taq polymerase enzyme. The enzyme latches onto the primer and starts moving along the template strand, adding complementary nucleotides, A, T, C, G, one by one, building a brand new DNA strand. These 3 steps complete one PCR cycle. And here's the amazing part. This cycle is repeated typically 20 to 40 times. With each cycle, the number of copies of the target DNA segment doubles. So after about 30 cycles, starting from just one initial copy, we can generate over 1 billion copies of that specific DNA piece.
This incredible power to amplify DNA makes PCR an indispensable tool in countless fields. Medicine, rapid diagnosis of infectious diseases like flu, HIV, COVID-19, detecting genetic disorders, prenatal testing. Forensics, identifying criminals or victims from tiny biological samples. Hair, blood, skin. Paternity testing. Scientific research, studying genes, cloning DNA segments, DNA sequencing, genetic engineering. Archaeology and paleontology, analyzing ancient DNA from fossils or remains. Food safety, detecting genetically modified organisms or microbial contamination in food. So we've seen how PCR uses precise temperature cycles and a few key molecular players to make massive numbers of copies of a specific DNA sequence. This powerful technique has opened up new windows into the microscopic world of life, helping us unravel biological mysteries and solve critical, real-world problems. I hope you enjoyed this journey into the amazing world of PCR. If you have any questions, feel free to ask in the comments below.